Shakti and Sitaprabhu for this exciting meeting. I think we all have the chance to share a little bit of what we do in our lab, and it's a small meeting, so every one of you have a chance to interact with the speakers since morning. And there is no difference. You can also stop me and we can have a discussion on whatever we are going to present today. First of all, in the back, I guess everyone can hear me, right? What I'm saying. So what we are trying to do in last 4 or 5 years, starting from when I started working in NBRC,
You have to put this.
this is something which is a very interesting area, and 2 areas actually in which we have invested a lot of time in the last 4 or 5 years. One which is dealing with essentially how does the brain actually develops when you look at the child or children, and what do we understand by when we talk about atypical development, when there is a different trajectory that brain circuits takes compared to actually a normal healthy development. The other side of the lifespan study is aging essentially. Some of the glimpse of that aging you saw in Sridharan Devarajan's talk in the morning, where he tried to talk about how does the underlying change in the structural informations may be associated with predictions of the brain age, right? So here we are not interested in brain age predictions for that matter. The behavioral data that is collected by thousands of labs across the globe are very murky already, with wide number of contradictory observations when it comes to actually developmental signatures, and particularly when it comes to social cognitions, is the way you interact with your friends, you understand each other's emotions, how do you mentalize actually somebody's humor versus sarcasm, or how do you mentalize those concepts at a very early age. That is exactly what is called as social cognition. The second thing is mentalization. Mentalization is a very important skill for us to become good learners by the time we hit school going age, right? And then the trajectory goes upwards because the brain areas essentially become specialized from a very salt and pepper structure, right? So during this typical development, children shows a very different kind of patterns of connectivity, and as our lab is interested in large scale brain connectivity, which is one of the way we think we can find the answers of what does it take to understand atypical development, and finding their biomarkers and specific set of applications in order to kind of develop interventional strategies to apply these to children who are actually suffering from mental health disorders. So what is social cognition and theory of mind? I have said already a bits and parts of it, but this figure already illustrates some idea about it. When we do something like Wisconsin card sorting, right, or shuffling things in right order, it requires certain amount of functioning from the prefrontal cortex, which are called executive functions in neuroscience. And if you look at actually children's, the first thing they understand when they are developing is how to sense pain, right? If they don't know what is cold versus hot, they may end up in an accident, right? So everybody who has seen a child in their house must have seen how children's perception of their own body and others' bodies, like hunger, pain, versus their internal states like beliefs, desires and emotions, change as you become older. And this is exactly which can be translated into word like I am leaving you versus something like who is he that is leaving, right? These are the questions which we ask actually in order to mentalize the concepts, or which are known as the concepts networks of the brain. Now there are 3 groups which has been very important and critical to understand this ability of mentalizations, or what is known as theory of mind, and it has been there for nearly 40-50 years and has been studied in non-human primates and as well as chimpanzees, because they also have this innate ability to understand each other therefore they can form social groups, right? Okay. So these insights about, as the autism cases are rising across the globe, including in India, okay, this becomes a very challenging question actually, that what are the hidden world of autistic children? How do they perceive the world? What is so different for them to perceive the world as compared to like neurotypical individuals? How do their brains structural and functional connectivity differs from each other? How do their brain dynamics actually differs from each other? So how a children's brain develop, you can also study using gifted individuals, gifted musicians, gifted mathematicians, and those groups can be also used as your study group or participants when you are comparing with the neurodevelopmental controls or typical developing controls. And the third group, which is of course the chimpanzees, right? Of course we don't do these experiments by going into the field, but some people do, right? So this theory of mind was coined by two psychologists, Premack and Woodruff, long time back, and these primatologists actually were thinking about these mentalization of the concepts. An example of these was an experiment which was done at MIT, where they actually asked these 4 years or 4.5 years old child in order to interpret things and response to others' thoughts and behavior. The child seamlessly learned this. Now a question comes, could an AI model can learn the same thing as the child does? How far the AI models will go in terms of overcoming what usually we learn at a very early age through examples, right? So the main questions that comes here then is, how do we perceive words such as we see or we look, and what is the meaning of see and look to a child who is at the age of 3 to 5 years? Words for physiological states or that indicates physiological states such as hungry, tired, emotional words such as scared or happy, and desired terms such as I want this, I want that, you want that, he wants that, right? How do we understand these words? What are the brain areas and their representations, right, in terms of perceiving these words and processing these sentences and comprehend them, right? And lastly, mind related words like belief, think, etc., etc. So we took a different directions completely. I won't say it completely tangential, but it's a direction in which our lab been working for a few years, which is where we are looking at the functional brain networks that we hypothesize to be responsible for recognizing others' mental states that are crucial for social cognitions or social cognition impairment, right? And if you look at the pyramid which is given by psychologists or classical psychological theory, they say that by the 0 to 2 years, the sensory motor ability actually develops. The children learns from crawling to standing, right, holding a support. By the time they reach 2 to 6 years, it becomes pre-operational, and the child uses symbols like words, images to represent objects, but does not do much of logical reasoning. Okay, the logical reasoning ability typically develops after 7 to 12 years, and then it becomes formally operational 12 years across adults. So the adolescents can reason abstractly and think in hypothetical terms, and therefore we think at these different stages of neurodevelopmental trajectory, it is possible probably to get a brain fingerprints in order to understand how does the brain connectivity is actually changing when you go from 2 years to all the way to 12 years and adolescents and all the way to adults. And autism spectrum disorders gives you a very interesting opportunity to investigate that, because there is publicly available data sets which is collected by many different sites, which forms a huge consortium data, which has all the information starting from genetics all the way to their behavior, right? So therefore you can relate almost at different scales how these complex information processing essentially goes through, right? Now, without boring you through psychological dogmas, I think what I will do is I will go next to, like, hopefully what happened, yeah, to the main question that we are interested in based on that background is how does the brain modules or the networks actually get reorganized when we look at typical versus atypical development, and any kind of atypical global modular organization, is it driven by node level changes? The nodes are brain areas in a graph theoretical abstractions, okay, because each of the nodes are the vertex of a graph and the edges are essentially their correlation strength. So therefore, we can actually look at their nodal composition in a resting state condition in the brain networks during atypical development and see to what degree it predicts actually symptom severity patterns that you observe in the clinical cases. Moving on, we can actually look at autism. The term itself comes from a word called auto. Auto means self. So it's a locked within themselves, removed from any social interactions, right? That is the name actually is coined by psychologists in order to talk about autism. It's a characteristics which is typically seen in two very distinct behavioral traits. One, which is impaired social communication. Second is repetitive and restrictive behavior, right? They show a very repetitive and restrictive behavior, just like the movie Rain Man, if you have watched it, by Dustin Hoffman, okay. And it's a disorder of functional brain networks, that is what our hypothesis is, comprise of these networks which are predominantly involved at the early developmental stages, including sensory motor networks that allows us to move our hands, that allows us to like do grip, touch and various other functions. Default mode network, which has come around late 90s by seminal works by Marcus Reichel and others, which has shown essentially the involvement of those brain areas in self-related processing, autobiographical memory and various other aspects. Then limbic network, which is also involved in terms of judgment of values and rewards, as well as like salience networks, which is a very important network comprised of mid-insular cortex and also part of the frontoinsular cortex, which is involved in actually maintaining a homeostasis in the brain, okay. And finally comes the control executive networks, which is composed of the frontoparietal brain networks, which I will show in few slides from now. I have lots of data, so nobody will go unhappy home, okay. And lots of maths is there. I have just skipped it because I have not shown it purposely, not that I don't know how to do maths, yeah, okay. Frontoparietal control network, and these frontoparietal control networks is bulk of what is required for decision making, such as moving left or right or going straight, right. So these decisions are controlled by these control executive networks, and this is a disorder which comprise of several of these networks, not just one brain regions or one network. So what goes wrong? Inhibitory control, which goes wrong. The control network do not send information to the sensory motor network most likely, as a result of which there is a very great difficulty for the autistic individuals to take motor commands, right, okay, or like understand the top down control or processing to stop their actions, right. So they keep on doing repetitively the same kind of things. Secondly, these leads to repetitive behavior, and thirdly, they are busy within itself, thus inefficient in processing the informations that is coming from the external environment or from the external world, right. So it's a locked in syndrome. But the question that we asked in this 2023 paper in autism research is, how does all these things fits into in the language of network neuroscience, and how can you exploit network neuroscience tools and methodologies in order to address some of these questions. So in order to do that, we actually moved further, and we put up 3 different ideas. So, the first idea was look at modular organization and composition in the age group, specifically focusing on 5 to 10 years, and the reason being, in the literature many people have looked at wide number of age groups. Therefore, like the resolution of understanding what happens in the early age groups, particularly school going age group, what happens became like little bit diffused in the process. So, therefore, we focused on a very small sample size in the age range of 5 to 10 years, because previous studies have reported there is a shift in the hub of the brains. And how many of you know about what is hub? So, what is the definition of a hub? Right. Okay.
Which connect most of.
So, I think the hubs are existing in physical systems, chemical systems, naturally occurring systems, and also man-made systems like electrical grids have specific hubs. Airport networks have specific hubs like Delhi, Bombay, Chennai. These are hubs. So, it can route informations to other cities in India, because there are a lot many connections from here as compared to a second tier city in India, right. So, those kind of hubs actually seems to shift from being localized in primary sensory and motor areas, which you will typically find in 5 to 10 years old if they are typically developing, to potentially more distributed patterns extending across frontal, visual, temporal and subcortical areas. These assortativity actually of the network seems to be very important, and the loss of this homogeneity of the network with early developments seems to be a very key to understand the autism spectrum disorders. It is a spectrum disorder, so you find autistics with very different types of variability in the overall functional connections. The second thing that we have looked at is the modularity of this connectivity. That means, do they form specialized units among the brain regions that controls the processing during spontaneous activity as well as during stimulus driven activity, right. And the last thing, we looked at the functional cartography of the nodal roles to see essentially whether some kind of hubs are changing their roles and they are becoming like kinless hub, or they are becoming something like non-hub in the entire process. Because if that happens, then there is a dramatic change in the overall network topological properties, and this change in the network topological properties or altered modular configuration arise from this atypicality of a node that is affiliated to a given module. Secondly, contribution of the node properties to this atypical module configuration essentially gives rise to a different set of neurocognitive networks, which gives rise to a differences in the processing of the sensory input. And then finally comes the correlation between these ASD behavioral traits and symptom severity scores to look for potential biomarkers.
So I have a question.
Yeah.
It's a mouthful of causal connection.
Right.
So your assumption is there is a problem with the hub itself.
Yeah.
Now that assumption, how do you know that to be true and the sole basis of driving this autism spectrum condition?
We have done some prior studies, but hubs are typically the most important units when it comes to the networks. Without the hubs, both anatomically and also functionally, and that's basically the primary starting point of any investigations. Okay.
Yes, but it is you are.
But we are not caring about the hub as such. I will show you the pipeline, then it will become more clearer. Okay. So you can look at.
I think one thing that just to comment.
Huh.
So that hubs are also, this is not like when you say
Causal.
It is not causal, it's also relational.
Yeah, because if the hub, like some kind of reorganization hub also doesn't guarantee autism, for example.
Yeah.
So that's the other way around.
So I meant causal means because the problem is being assigned to hubs, and then the behavioral inconsistencies being
Yes, so it's like a way to predict the symptoms more. You can think
Yeah.
of it as that.
Yes, but I am more concerned about the presumption that how is the.
So two things are there. One, there is no causality here. Okay, we are pointing to. Second, these are basically the observations that is coming from the network level analysis, right? To see essentially what is the altered network topology in autism and whether that has association with the behavioral scores or the symptom severity scores that is typically taken in clinical settings. Okay? Yeah, so there is no causality here. I am not talking about any mechanisms right now. So, in order to look at the modular organizations, typically if you look at the methods, normally you will do essentially a consensus agreement either using Newman-Gidwan algorithm, which was published many years ago in PNS, and also like you can use Leuven spectral clustering methods, which is a greedy optimization methods, and using that you can essentially get the community structure of subject 1 or subject 2, and you can extract two types of information. One is the modular composition and connectivity, and second is basically to get an handle on the local node properties in that particular graph, right. But these are not sufficient to give you the understanding of the module affiliations. So, therefore, there is a separate metric which was derived by the computer scientist, which is also called neuromodulation index, and these normalized mutual information is that neuromodulation index, which actually gives you the confusion matrix to calculate essentially the community structure of different individual subjects, right, by using the partitions to find out essentially what are the scores or values of NMI, which is a normalized value between 0 and 1. So, if it is perfectly 1, then you know essentially two of the communities are self similar. If the score is equal to 0, then you will know that the two communities are completely different from each other. So, using that you can essentially determine the community structure, how much it has changed in those children in the 5 to 10 years age compared to like the neuroatypicals, right. And then you can actually look at their group level differences between autism spectrum disorder and also typical developing individuals, and that is what we looked at both for the left hemisphere and also the right hemisphere. So, you can beautifully see essentially how the community nodes or the nodes that belongs to a particular community has sort of clustered together, right. And when it comes to like autism, some part of that cluster remain unaltered, okay. But there are many networks or there are many brain regions which actually change their module affiliation, and therefore that actually seems to change the altered topology or the backbone of the network that is actually present in the autistic group compared to the neurotypical individuals. We also looked at second measure, which is called nodal cohesiveness, that if it is a visual area, how many regions of the visual area, which is known from the literature, is part of the visual module versus how many of them actually now got affiliated to another network. So, this nodal cohesiveness, as it actually increases, it seems that the mean participation coefficient decreases, and it is not counterintuitive because participation coefficients measures in a given network how much is the integration between brain areas, okay. So, if you have lesser integration between brain areas, okay, then you have higher nodal cohesiveness, that means the nodes are belonging to the same module and it has not changed its community affiliation in the course of atypical neurodevelopment. Whereas, if the nodes have changed their community affiliations, the participation coefficients are actually pretty low, almost close to 0.1 or so. And this is the case essentially for both ASD and neurotypicals, that you do find there are certain differences in the nodal cohesiveness to a module, but far more in the case of the ASD, and which is primarily located in the default mode network, sensory motor network, salience network, and the dorsal attention networks. And these are the brain functional networks I started with, actually describing that the core symptoms of the autistic deficits actually starts from some of the brain areas which are anchored by these networks. When we looked at the intermodular connectivity or between network connectivity, what we found is essentially that within and between network connectivity between the ASD and also the TD actually is substantially different at the group level, and the differences come from the connectivity between DNN and central executive networks. That means the self related processing areas of the brain connected to the executive functioning areas of the brain, as well as sensory motor salience and sensory motor and salience networks and central executive and sensory motor networks, suggesting that autistic individuals have more hyper connectivity, okay, as compared to essentially typical developing individuals in some of the brain networks, whereas they have more under connectivity or hypoconnectivity in some set of other brain regions. Okay. And these hypo and hyper connectivity actually makes them or makes their world probably very different, but that is very speculative till now, right. So, with that background study, we moved into writing a review article with one of the prominent figure in autism research, Lucina Uddin, and Lucina just came up with a paper yesterday in Nature actually, which is called Resting State History and Future, right, with Bharat Biswal, who was the one of the guy who invented resting state fMRI. And Lucina and myself, who is located in UCLA, we wrote this review paper. We were very unsure that how much information we can claim or how much evidence we can collect in terms of core versus peripheral brain areas and their brain dynamics in autism, and how much information actually exists, how much people have actually done this type of research or work in order to going through their data. Fortunately, we found some core periphery interaction papers which actually also came through email communication with Henry Kennedy, who actually gave the idea of the diffusion tensor imaging and the connectome idea. So, they also shared some ideas previously about the core versus peripheral dynamics of the brain networks. I will just quickly summarize that. I am not going to go into the details of it. In offline, if you want to discuss, I am more than happy to discuss obviously about it. What is happening is that we have seen at one hand at the nodal level, the nodal cohesiveness changes. This has an impact on the participation coefficient and the overall integration between the functional module. The modular composition also goes down, and the modular composition becomes more erratic in autism as compared to being more cohesive in the case of typical developing individuals. Here what we find from a completely different analysis is that the flexibility of the networks actually changes, particularly the flexibility of amygdala, which is responsible for emotion processing, particularly fear and anxiety processing, as well as thalamic nuclei, putamen, and superior temporal gyrus, which is part of the speech and communications. These are the brain areas which becomes more and more vulnerable, but some of the brain areas also become less flexible with age. Okay? So, they become actually like less flexible in the sense that they become actually rigid, there is no change in the community affiliations, and there is a overall stability in the network, and that stability in the network seems to be very indicative of the autistic disorder of these pervasive symptoms, right.
What's the time scale over which you are looking at these correlations?
So, these flexibility scores we are estimating from the functional MRI time scales, which is in the length or durations of typically 10 minutes or 600 seconds, but then each of the correlation matrix is actually averaged over 30 seconds, over a sliding window of like 30 seconds or 20 seconds, because you first actually estimate the dynamic functional connectivity from those sliding window analysis. So, you essentially take overlapping or non-overlapping windows, and from there you can actually estimate the neural flexibility. Okay. So, these are rather longer time scales as compared to what you will find in electrophysiology or like EEG or MEG signatures, but these dynamical functional connectivity that you estimate, from which you infer the neural flexibility of the modules, seems to be like frequently used in the fMRI literature, because you cannot go down in a smaller time scale than that. Then you start actually seeing lot of transient activity which does not provide you a stable configuration. Yeah. So, later
I have another question. So some of the features you were talking about
right
are also can be caused by early life stress.
Oh yes, yes, absolutely, absolutely, very important question actually. So, there are certain things that are controlled for. So, for example, the autistic individuals or autistic children usually have a 4:1 ratio of male versus female. So, most of the autistic data that you find is predominantly male data. There are lesser female data. There are also like differences that can come from the lifestyle factors, and there are also differences that can come from like stress they have received, or stress that is like introduced at a very early stage. Yeah.
Selective retention of say or less pruning happening in certain brain areas.
Right.
That can like amygdala if you take.
Huh.
Someone who had
Right. Right.
early life stress
Right.
can have an hyperactive amygdala.
Absolutely, it can. The only challenge over here is to kind of regressing everything to mean, because autism spectrum disorder is a wide variety of spectrum. So, you can find those kind of individuals, but then you can find their aberrations as well. Yeah. So, the interim summary is functional hyperconnectivity and increased between module connectivity seems to be topological reorganizations that is observed in the time scale of fMRI data, at least in the atypical neurodevelopment compared to like typical neurodevelopment, and there is a reduced alteration in node community membership, particularly in 3 functional network: default mode network, sensory motor network, and salience network. These 3 networks has been widely reported by at least, if not less than hundreds of studies in autism, in mouse model, in human, okay, and also in other species. So, therefore, it gives certain metal to our finding that these probably we are hitting somewhere closer to those areas which are showing actually altered modular composition as we develop. Okay. And yeah,
You select the data is from the avoid data, right?
this is from avoid. Yeah.
So when you select the subjects for your
Right,
analysis,
right.
So did you choose any kind of way to constrain these behavioral symptoms? That
Yeah.
you didn't take randomly any autistic children, but
Yeah.
you took with certain behavioral symptoms. Did you pay any attention?
Yeah. So, particularly they had these ADOS social
Right.
and ADI scores. So, for those for which FIQ values, ADOS total, ADOS social, and ADI, speech and communication, was available, only those selective subsamples were taken
But
into
you
these.
took it over a standard deviation of those.
Yes,
numbers, so
yes,
that score
yes,
some range.
right, in a
And
range.
would you consider them falling under like what category of autism?
These are all like high functioning
High
autistics. They are like, there were no low functioning autistic
So
children.
will that result hyperconnectivity change in low functioning autistics like the savants? Or?
We did not look at it, right, because we only focused on high functioning autistics, primarily because each of those centers or sites only reported some of their data or full data, and some of the other individuals with low functioning individuals particularly, they could not cooperate throughout the experiment, right. So, they had to leave out them at some point of time, right. That has been of course one of the great challenge in autism, to look at a specific set of subgroups or samples and infer about population, right. So, these I will just bypass. So, the next question comes is, if all these are so important, why do we talk about dynamics? Are they anything meaningful, useful for us to look at? Because what we have looked at is essentially a static graph with alteration in their modular composition, right, their community structure, and based on that we have inferred about the topological reorganizations that is happening in the atypical group, right. But while we say all these things, one thing that emerge actually as a very important methodology is basically the temporal changes in the brain network or time resolved changes in the brain network. And this is very important because cognitive processes are in general very dynamic processes, as has been presented by seminal works by Medaglia or Danny Bassett, right. Danny Bassett and others have shown essentially that these cognitive processes changes in memory, learning, attention, fatigue, and all types of condition and context. Secondly, these network structures actually change over time. So, the interval of time of observation, and the question what Collins also asked, is very important, because as you change the temporal window, the network starts evolving, right, to a different configuration. So, the question that comes is, how does, with different tasks or as a function of learning or as people are becoming older or with the progression of the disease, this network actually evolves, and is there a common methodology that can be adopted in order to address some of these questions, right, in the context of the problem that we are interested in. Of course, it can be applied in many different domains of applications. So, we reviewed the literature and we found very interesting set of studies which was reported by Henry Richardson and Rebecca Sachs from MIT, where they looked at the functional connectivity as a feature of healthy participants, about 155 of them, during the development of the theory of mind brain networks. That brings us back to the theory of mind brain networks, which we started with, important for social cognition. Then there are many other studies, including the study by Aglin's-Kass, which was published in Science in 2022, where they looked at the same data set which I was reporting about, but a much larger sample obviously of healthy and autistic individuals, and looked at the structural imaging differences in those individuals to find out the common source of variability between autistics and neurotypicals. And finally, Kostov Supekar with Vinod Menon from Stanford developed this technique using dynamic functional connectivity and generalized neural network models to do classification of ASD samples on a rather very large data sets, which was published in Biological Psychiatry. So, using these background studies, we asked, all these studies are good, but none of these studies actually looked at the really quantification of the task fMRI data. So, why not look at the task fMRI data if we have the data even from the normal healthy children, not necessarily from autistic individuals. So, we targeted 3 to 12 years old children's, about 122 of them, and 33 adults as our controls, and then we let them watch a cartoon movie in the scanner, right, functional MRI scanner. And this was the data which was collected at MIT by Nancy Kahnwischer and also Rebecca Sachs with their postdocs Henry Richardson, and they did another task which is called a theory of mind task or a false belief task, which was performed outside the scanner, okay, once the scan was completed. These are the results. So, when we plotted the data, we found that these are the brain regions, the left temporoparietal junction, right temporoparietal junctions, which are known as the area for variety of cognitive functions, including reorientation of attention, social cognition, as well as understanding the belief of others. In addition to that, they were main areas of the default mode network called precuneus and also ventromedial prefrontal areas plus dorsomedial prefrontal cortex and posterior cingulate cortex, which responded to this stimulus. So, the stimulus were Pixar movies, cartoon movies like Toy Stories and those kind of movies, and the children has to understand that there is this character called Gus, but this Gus is a cloud, but there is a character hidden in that cloud. So, if the children understands that, that means their ability of the brain networks has developed significantly well in that particular age, so that they have an emancipation and understanding of to conceptualize these idea, right. And similarly, it went on for the other stimuli. There were about 5 stimuli which were very well labeled, and these were called scenes of the movie or clips of the movie that is related to mentalization of higher order concepts. These were the scenes which are related to mentalization and understanding of the body pains, right. Yeah. So, these are pain scenes which are labeled as P1, P2, P3, P4 and P5. These are pain, so that the understanding that the children have a good pain perception, right, at a very early age is developing, we can understand from there, right.
Wouldn't it be a better idea to feed them something that they have never seen? Because
They have not seen these.
How do they know?
Yeah.
It's a
Yes.
Pixar movies, right?
They have not seen these, the children's.
So in the US, at MIT, people they are
So, they have not seen these, they have included, there is an inclusion criteria for the participants who have never seen these movies. Okay. When you do a study in children in the age of 3 to 12 years, you also have to take the parents' consent, right. So, the parents also actually go through a series of questionnaire which are usually used before you recruit and include somebody for fMRI acquisition. So, you are absolutely right. So, one of the question was there was no repeated exposure, right. They have never seen these. Okay. Otherwise, what will happen is these bold signals that you see, where you look at essentially the normalized signal, that will go down because of the novelty factor is gone, right. Yeah. So, there is an effect which is called repetition suppression in cognitive neuroscience. So, that is exactly what will happen if we plot the amplitude as a function of scan number. Yeah. But here we found those areas which are responding at a particular time interval or time windows when they were exposed to these scenes, and certain movie clips evoked higher activations as compared to other movie scenes which were not interesting for the children. So, these are basically the description of those movie scenes. If you want to do a different type of analysis based on, let us say, large language models or something like that, where you feed essentially some kind of semantic inputs, right, you can do that, but we have not done that anyway. But we looked at basically these brain regions and their MNI coordinates, specifically which depicted essentially the functional MRI activations. Now, once you have that, you can go for creating a new method which is used in estimation of dynamic functional connectivity, where you can choose a windowing scheme, which Collins was asking also. These are the time windows, right, over which you see this time series which is coming from different brain regions, which is often used in time series analysis, and over these sliding windows you can estimate the matrices in time and couple each node to itself in the following matrix. Okay. So, you get essentially a windowed functional connectivity, and you can go further, you can estimate what are the brain states, how long an individual stays in that particular states before it jumps to the next state and so on and so forth. Previous studies have shown that these jumps are important, or these state transitions are important, because they can be related to the working memory performance or cognitive flexibility of the individual subjects. So, therefore, the brain dynamics and their transitions can be very, very meaningful in understanding the transient states as well as their relationship with cognitive flexibility. Again, this is not causal, right. This is only correlational in this case. So, what we did once we had the data is take a different approach than what exists in the literature as is depicted in the previous two slides in terms of windowing the data and analyzing those correlation matrix, right, based on the sliding window correlation. Instead of that, there was a PhD student who was working with Arpan and myself, and we published a paper few years back in cerebral cortex where we took the same data and those time series signals and we tried to estimate their phases. The phase is depicted as theta, n is the number of brain areas, t is the time, and the cosine differences essentially gives you how different they are in terms of their phase angle, right. If the phase angle is 180 degree or 2 pi, there will be a huge difference between the two, whereas if the phase angle is actually 0 degree, they will be very close to each other. So, this is a measure of what is known as also angular distance measure. So, you can estimate these DFC using these cosine values, and these are the leading eigenvectors which can be projected to the dominant dynamic functional connectivity subspaces. So, we ask the question, if we compute the dynamic functional connectivity subspaces, all these codes are available online. So, if somebody wants to use these codes in order to figure out the DFC subspaces for any functional MRI data in which you are working with, please feel free to use it. Okay. So, you can calculate the dominant DFC subspaces, and from the dominant DFC subspaces you can measure the angular distance and also another metric which is known as Mahalanobis distance. Based on that, you can ask actually, if we look at the temporal stability of the pain processing areas of the brain, because pain processing scenes were there in the movie, versus theory of mind clips that was processed by the children, would we see actually a difference in the underlying subspaces, okay, in any specific way, which will indicate that the temporal stability of those functional brain networks actually gets changed or altered in a dynamic way. We had also looked at the behavioral data. It seems by the age of 7 years, all the children are able to pass the task that was done outside the scanner. Okay. So, they will go through a mentalization task or a theory of mind or a false belief task, and in that task they were able to reach high percentage of correct response or answers. As you can see over here, it is touching the plateau of 1 and they were perfect. So, which means that somewhere around this age 7 years, the children's ability for mentalization has reached this level which is comparable to the adults, right. Okay. And we wanted to find out whether our data from the functional connectivity measures of angular distance, which depicts temporal stability, can actually capture that. So, you cannot see over here because this figure is not zoomed in, but these are the brain areas that we have already talked about, that was used during the movie clips or movie watching task, which showed higher activations that I showed few slides back. So, it composed of precuneus, ventromedial prefrontal cortex and various other areas. So, when we looked at the bold-face coherence, that is the cosine of those differences in the angle from the time series data, for the instance of the movie where they had the pain processing clips or theory of mind processing clips, we found there is a distinct difference between, like, these temporal windows, which means that the Tom versus pain scenes are processed very differently by the children's, and the temporal stability of the respective networks are also, like, turn out to be developmentally changing, going from 4 years old to 5 years, 5 to 8 years to all the way to 12 years. Okay. And the next observations that we had that this phase coherence actually is a very important measure, it turns out, and it actually kind of gives you the temporal stability metric which you can derive all the way from 3 years to adults, and by the time like you reach about, let us say, 5 years or 7 years, you can see that the pattern between the 7 years and the adults actually start showing substantial similarity, right, which was the point that we were making, that when they were doing the behavioral task outside the scanner, by the time they reached 7 years, they were all giving kind of accurate responses. We did not stop at that, we moved on and tried to predict those children who passed the task or failed the task or gave inconsistent response in the false belief task, whether that can be predicted from their brain connectivity patterns. And it turned out that the pass group's brain connectivity patterns between those set of brain regions is very different from the fail group's brain connectivity patterns, and the brain connectivity patterns between the same set of brain regions were mostly weaker connectivity as compared to the stronger connectivity as you see in the pass group. And in fact, the inconsistent group which we found seems to have a connectivity which is somewhere in between the passers and the failures, which is something which you will probably expect. So, I think the key findings were temporal stability of dynamic functional connectivity is meaningful. It is not only that the static graph topology and the altered modularity and modular composition is good enough sometimes in order to actually look at the inferences from the developmental neuroscience, but in a complex scenarios like processing other bodies or internal mental states of other minds, it requires actually more than static graph theoretical network methodology, and to embrace these dynamic functional connectivity analysis in order to look at the ever changing patterns, right, in the brain. Secondly, we also found that the false belief performers usually maintained a lower distance in the angular distance measured, and higher repeatability in patterns were observed for passers, with increasing distance and instability observed for those who failed. Now, this is very interesting because we can take these data and observations which is coming from the failures when we have a similar task carried out in autistic group, right, because if the autistic children of the same age and gender matched are not able to process the same task, then one may wonder whether the connectivity will turn out to be similar as you observed in the case of the failures, or would it be any different from the typical developing individuals, because these are still typical developing individuals. And finally, this further suggests that increased functional correlation in the TOM network may drive the stability of DFC patterns and may be associated with age, as we saw that when we moved from 3 years old to all the way to 12 years or adolescents, the connectivity got more and more stabilized over time. So, I think I will just stop over there. Just I will show you this paper which came out 2 days back in Nature, which is a review article, not an original research article, but it talks about the importance of machine learning in precision psychiatry. On the same idea that I was advocating throughout my talk today, that you take a sample, you do not know, the sample is very salt and pepper, right, who is autistic, who is normal, who is not. Sometimes there is not many clarity, good clarity on that, although there are clinical diagnoses that are very much like routinely practiced in medical facilities, right. But if you take a sample, you can quickly go through the training process, you can also kind of divide these into a certain percentage of data for testing purposes, and you can develop ML models and extract these resting state fMRI features and use ML model to estimate phenotypes of interest. In fact, you can relate the phenotypes to the underlying genotypes of these individuals to find out developmentally whether there is any kind of change in any kind of gene expressions, right, based on transcriptomic analysis of the Allen Brain Atlas data, and you can relate these phenotypes with the genotypes that you find from the Allen Human Brain Atlas and see whether to large extent they can be associated. They are still not causative models, they are still correlational models, but this is the best you can do at the moment, right? And then you can go into derived resting state fMRI features. You can extract independent component analysis, find out the voxel wise activations. Voxels are typically of the size of 2 millimeter by 2 millimeter by 2 millimeter. It's a 2 millimeter cube over which you can spatially resolve the activity using fMRI. That is very as good as you can get in terms of the spatial resolution. So you get these activations, and then you can look at the whole brain functional connectivity. You can apply graph measures like the ones that I have discussed over here, and then you can use like machine learning model optimization by extracting recursive features or like supervised ML models, or use like very routine classic support vector machine kind of ideas to explain the boundaries or separation of the boundaries to find out like different types of clinical subtypes, right? Sita bro, am I out of time?
No.
Do I have 5 minutes?
Yes.
Is it okay? I think I am competing or trying to compete with my previous speakers. No, sorry, I will stop. I think I cannot be as funny as Collins, right? Okay. So just wanted to tell you that this, in my opinion, although gives you a very good methodology and pipeline to go after, given the current set of or current volume of data, I am not still very comfortable with simple models like these, because there are much more complexities and nuances that need to be addressed. Okay, so one has to be very careful on both sides, on neuroscience data sides because it's extremely noisy. On the machine learning side, we understand those deep neural network models really less and less, okay, although they show a lot of success. So on both sides we have to go through a conserved reserved approach, okay. But what definitely is needed is to look for a model or a framework that looks at beyond pairwise correlations, because what we have been talking about till now is actually looking at pairwise correlations, right? Okay, we take region of interest A and B and we look at their covariance matrix or correlations based on linear correlation values, which is mainly the Pearson correlations. But there is lot more when you go into the higher order spatiotemporal organization and look at neural network models, and this has been this came out fairly recently in 2023 by Lucas Arbabids and Spase Petkowski, and they suggest that this higher order spatiotemporal organization of the dynamic functional connectivity that I talked about in the previous slides are important for clinical neurodegenerative as well as neuropsychiatric symptoms. So what we did in a very recent work, we looked at 3 types of connectivity. The first connectivity is your pairwise correlation between brain regions, which simply gives you static functional connectivity. Second, from the same data, we looked at the estimation of the dynamic functional connectivity, which actually is like the sliding window approach that we have talked about already, to extract the temporal correlations. And finally, we went into more complex metrics, which is called actually edge connectivity. Edge connectivity means you take the edges and you cross correlate between the edges to find out the trimers and the dimers, right. These are also called triplets and quadruplets. So you can look at higher order correlations between the links. So this is like a link to link correlations, or sometimes it is also called spatiotemporal correlation or meta connectivity. Using this meta connectivity, we have proposed actually graph neural network models, because we have a graph representation at the level of static functional connectivity, dynamic functional connectivity, and also meta connectivity to do some autism classification, right, starting from some samples which is drawn from the populations. And then we have the graph representation. So, the graph ages are learned by these model. This model is called knee-gut model that we have actually proposed. And in this model there is an attention module which actually gives us an idea that how those connections or ages, or which connections or which ages contributes most to the prediction. Okay? And in this process we can find that we can get the meta connectivity values for each subjects, and we can predict actually the brain to behavior association very nicely and perform testing also on the unseen data. So, the graph model that I showed before, it can learn actually the features based on the age correlations or meta connectivity correlations. However, this is a higher order feature, so it requires a lot of computational time. So, in order to reduce the computational cost, we had to actually fundamentally break these networks into two different modules. First, we had to go from graph to subgraph and then train the ages or age correlations on the subgraph in order to do the classification task. And in this process, we can do both things. We can do the classification task to classify between ASD and neurotypicals. And secondly, we can also make use of these connectivity matrices of the participants to predict their behavioral scores, which is coming from the clinical setup, right. And finally, we can predict the brain areas which are responsible for these symptom severity patterns. Of course, many of the clinical scores which Arpan also asked about, like ADOS total, ADOS social, ADOS communication. ADOS is basically a scale though. Using that scale, which is a standardized scale actually, the clinicians usually measure their speech and communication ability, or to what degree their speech and communication has impaired, or what degree their social cognition has impaired. So, those scores are available for these subjects. So, you can make use of those scores in order to predict how much these connectivity ages positively correlates with the scores, and those ages which actually negatively correlates with those ages. So, final conclusion is meta connectivity and attention based graph neural networks give the best performance in classifying autistic individuals from neurotypicals. It surpasses what has been predicted in the literature based on a handful of studies using either static pairwise functional correlation or using dynamic functional connectivity measures. So, it seems there is a importance of going beyond pairwise correlations, looking at the higher order spatiotemporal correlations, because it may be having a very good diagnostic value, even though it may seem very complex at the beginning because of the way it is estimated. And these three subgroups we identify, I have not discussed about it in ASD, which is in the severe category, moderate category, and also in the mild autistic category, based on the meta connectivity patterns of their individual brain regions. And the connectome based modeling approach using meta connectivity gives the best performance for clinical symptom severity scores. So, possibly it could be extended in a future work to develop some diagnostic tools. These are my funding agencies and also some of the collaborators and students who primarily have contributed to this work, and at least one or two PhDs which came out of this work, and as well as the collaborators from outside India. So, thank you for your attention. If you have not asked questions because you are afraid to ask or shy to ask, please feel free to ask, because this is a workshop which is supposedly kind of interactive as I understood. Yeah, please.
So, some of the you have healthy group and
Right.
now these healthy children.
I would refrain from the term healthy. but I will call like more like typical developing group. Typical developing group. Although within typical developing group also there may be a lot of variations, right?
So the control
Yeah.
group,
Yeah.
children from the control group
Correct.
can later be reclassified to be in the ASD group, right? And the vice versa is true.
Possible, but mostly autism, at least the extant literature suggests that autism gets detected at a very early age normally. Okay, so if someone is not found autistic by the age of, let's say, 10 or 12 years, it is highly less likely that this person will become autistic. But that does not say that it might not happen. Under certain situation and circumstances it may happen. Just like there are studies in schizophrenia where people have lost their home because of migration and other problems, and because of those new environment where they had to settle in, that isolation caused and triggered schizophrenia. So these may happen, but the cases are really small in terms of the number of children who are detected as autistic at a very early age. In fact, that is one of the biggest problem in autism, is to detect somebody as autistic at a very early age, even before the school going age. Often they get diagnosed after they enter their primary schools.
So does it suggest that there are definitive genetic components?
There is a huge genetic components. At least lot of animal models and studies does suggest that there is a a neurodevelopmental disorder.
There will be genetic component, yes, but genetic component alone does not predispose or in any way.
Yeah, that is for sure. There is a nature versus nurture debate also there. Okay, so while the conditions, okay, under which they,
Polygenic also.
it's a polygenic also. There it may run the risk in the family also. We have not looked at those type of data where you have siblings, for example, right?
That's why there are maybe 50 genes are responsible for giving up.
Right. I did not go through.
I am concerned about the control group because everything you have done is based on the authenticity.
What is your concern about the control groups?
I am not concerned in a way. I am not certain whether the control group actually constitutes the control group.
Yeah.
Because if there is some discrepancy in the control group, all your correlations have to.
So that control group, I think, comes from that ADO score. So that's the gold standard.
Yeah.
So that ADOS, there are
So-
certain repetitive behaviors which are very common. So these will be there present in most autistic child. So one is lack of attention to eye contact.
Right.
They would not make eye contact typically. Second, typically following emotional content in the stimuli. So for example, they don't understand typically you say something like do this versus do this. So the difference between a very authoritative sort of a
You are defining me actually.
so the point is that a lot of these child, they actually don't distinguish between the two. The other sort of which actually Divanja didn't mention much, but a lot of us we believe sort of autism is not. This is where I differ with Divanja, I guess. So it's not a disease of higher order cognition. So it's a disease of sensory disability. So basically, there is if you think about your neural system, there is inherently a lot of noise in the neural system. When the signals are coming through the primary sensory cortices, there is essentially more noise in their
You could
system.
organize our own stock now, right?
So more noise in their system, which kind of contaminates your development to other sort of higher order forms. Like for example, the concept network. Which so what I'm saying, so everything is very loud for them. So when you are when they are dealing with sort of, if you listen to any kind of sound, small sound,
Hypersensitive.
so they are very sensitive to sound. For
So
example,
typical developing group, as what Arpon was saying, it's not just based on ADOS scores. Okay, typical developing group is identified based on maintaining certain amount of cognitions there are different cognitive measures, there are fluid intelligence measures, there are IQ measures, lots of different measures have been taken by people to essentially identify somebody as a
DQ, IQ
typical
comes at a
developing,
later stage,
yeah,
but DQ.
yeah, and also emotional quotient, right? Emotionally they are very vulnerable. They don't understand the difference between like humor or sarcasms. Then they have these repetitive restrictive behavior which would normally won't be manifested by any individuals. They would like, for example, their lunch to be packed in a certain way and every day in that same way. Okay. So these kind of traits, behavioral traits or personality traits are not exhibited by typical developing individuals. Okay. And this is also the case in case of like when you are looking at mouse models where there is a gene called MECP which gives rise to what is known as Rett syndrome. So if you have studied some of the papers by Mriganka Sood, he has identified essentially a specific set of gene called MECP gene which is responsible for the Rett syndrome, which also shows these repetitive restrictive behavior in mouse and severe deficits in social cognition with another mouse.
Yeah, so they keep on.
Correct.
Even if they bleed, they keep on.
Right. Even there you need control, right?
So we just take 2 more questions because
Yeah.
of the lack of time. So one here.
So I hope I was able to at least tell. So yes, please.
Continuing with his question, sort of.
Yeah.
So empirically, a lot of people like I know and know of have been diagnosed with ASD pretty late in their life, like early adulthood. and all.
Right.
So and since I mean you're right that like symptoms of ASD develop early, but for some reason they have been diagnosed late in
Right.
life. So how are you so sure that the control group does not include such individuals? Because like the behaviors that you say, like obsession with organ.
There may be a possibility, but the number of samples, if you see, we are analyzing, is more than 1500 here. So if there is 1 or 2 cases also like that, it would be very difficult to detect that, first of all, and secondly, I don't think it is done just like that you leave it to chances, right? So
Done in clinical settings, right? So they must.
can I finish my answer?
Sorry.
Okay. So essentially when you are doing these studies, fMRI studies, you are doing scan on any studies fMRI you do. That's why first slide, if you remember, I talked about gifted people versus not. So if you are musically naive versus somebody like A.R. Rahman comes, there will be obviously a difference in the brain connectivity patterns between those individuals, specifically those areas which they rehearse every day. So our resting state connectivity is reshaped by training. So if the autistic individuals are going through the same routine and repetitive and restrictive behavior, we are just saying essentially functional connectivity is one way of differentiating between autistics and the neurotypicals, right? So I gave 3 examples. One is you can do the same experiments in non-human primates. You can do it in gifted individuals versus not gifted individuals, okay, controls who are not musically trained or who are not trained in mathematics, for example, or you can do these in autistic group versus neurotypicals, right? So I am interested not in autistic, I am interested in how does the brain is developing typically versus atypically, right? Yeah. And what does allow the brain to essentially acquire cognitive or neural flexibility, right? So if you cannot pay attention, you will fail your exam, right? If you cannot stop yourself when you are crossing a street, you will be in the danger of hit by a car. These are basically the ability which autistics will often not exhibit, which are called inhibitory controls, executive functions, and those kind of things.
I think with some of the definitions regarding autism are a bit loose.
No, not really, not at all. I would say. Autism term actually came from self-related processing. Okay, so you don't understand who you are, and your internal concept, your internal reference frame is distorted actually. It's a disease of default mode network actually, I would say rather. I can show you some.
It's a sensory process.
No, I wouldn't. I will show you result if you want We see these are the genes which are involved in autism. We have also analyzed that. These are the brain areas. So, Arpan is wrong first of all. Okay. So, there are a lot of areas which are involved which has hyper or hypoconnected genes, ASD genes, including motor cortex, temporal pole, superior temporal gyrus, primary visual areas, inferior frontal gyrus, inferior parietal gyrus. So, you can see over here there are top 10 highly expressed ASD genes in those same areas which I was talking about. So, I did not have time to talk about these. In fact, these neurotransmitter specific functional activations are all over the place, which hinders in reward processing, dorsolateral functions of face recognition which Arpan was talking about, right. So, these are
Basically everything the brain does.
no, those which are in bigger words,
Okay.
these are the more deficits in those domains. Those which are in smaller words, like for example ventral visual reward, or those ones which you do not even see, right, those functions are not impaired in autistics. But the primary functions related to motor ability, the primary functions related to face recognitions, these are highly impaired. And if I show you the last slide, you will be even wondered what is happening in autism is that the information which Arpan was talking about, if you do a functional gradient analysis, you find there is a gradient difference between neurotypicals and ASD because some of the information gets stuck. There is a bottleneck in primary sensory areas, and these information never reaches actually to the default mode and other areas. So, you have all kinds of evidences in the autistics.
Absolutely correct. So this
Huh.
is what I am saying.
Huh.
So that this sensory information is not reaching out.
Right. So, this is where many people suggest that the sensory information goes through a bottleneck. There is no information update, where these informations that comes from the environment need to be relayed to the next layer or next area of the brain.
That is essentially make it an EI balance problem.
It is also a EI balance problem. You are very right about it. Of course, I have not shown any data on EI balance at the moment. Yeah.
For the sake of
Yeah.
one last question.
Sure. Sorry, I took longer route to.
I actually didn't have a question.
You wanted to comment actually.
Yeah, I
Please go
wanted
ahead.
to comment. But you actually covered more than what
Did I
was
cover?
saying.
You can bring new ideas.
So what he was saying about
Huh.
early life stress can cause hyperactive amygdala.
Right.
And if in the experiment, if that was considered as exclusion criteria or not. My theory is that we for sure don't know what are the causes of autism as of now. Like there are speculation that the genetics is involved, but many other things are involved.
Right.
So excluding based on early life stress won't be correct criteria because it might be the causing factors of autism itself.
So he said about one such factor that can
Yeah.
trigger autism. There could be multiple factors, and the other things I want to point out on his point, he talked about loose definition of autism, okay, or loosely, autism is loosely defined. So autism sometimes is actually a cluster of disorders. So an autistic can also manifest other types of symptoms. In fact, there are studies which have looked at both attention deficit hyperactivity disorder at a similar age range and compared that with also autistics to look at essentially if there are any functional connectivity differences between autistic individuals versus ADHD, or to what extent or what degree they overlap, right. So, it's also can be comorbid with other types of disorders as well.